So yes, today I'm going to be talking to you about a continuation of the Tapestri workflow that Gema started last week, where she gave us detailed information about encapsulation, barcoding up through targeted PCR. And today my talk will be about the next steps that take place generally on day 2 of the protocol, library cleanup, library PCR, and then some sequencing considerations. So here's the agenda for the talk. We will first go over breaking of emulsions, which is the first step after you get your targeted PCR product from the thermocycler. We will then talk about the cleanup steps. We have a 2-step cleanup process. I'll go in detail of how we do that. Talk about considerations for your library PCR for multiplexing multiple samples. And then we'll talk about sequencing considerations relevant to your Tapestri library. So the first thing I want to point out is that everything you do on day 2 of the Tapestri protocol actually uses equipment other than the Tapestri itself. You don't need the instrument depicted here any longer after day 1. All of those steps take place, encapsulation and barcoding on the instrument. Subsequent library prep steps take place with bench equipment, vortexers, centrifuges, and notably a few that I've pointed out here. This is from our single-cell DNA protocol. You do need a type of capillary electrophoresis method, generally bioanalyzer or tape station. Others would work fragment analyzer. Some form of DNA quantitation other than just looking at the distribution of the library. We recommend qubit. You will need a thermocycler that is capable of supporting ramp rates from 1 to 4 degrees per second. Many of these are available on the market. Probably you already have these in your lab. There's no set one that we recommend, but we can work with you to help you establish the correct thermocycling protocols. We'll also need a couple of different magnets, and I'll go into detail about which ones you'll use in which steps of the protocol. And then lastly, all of what I'm describing now is upstream of an Illumina sequencer. Any Illumina sequencer will do. And in the future, we'll have additional capabilities. Right now, everything is libraries that will be sequenced on the Illumina platform. And I'd like to point out that we have at Mission multiple single-cell protocols for looking at DNA and protein together. We call our multi-omics workflow. We also have protocols for looking at dissociated solid tumor cells and nuclei isolated from such cells. Those I'm not talking about today. I'm just really focusing on the standard single cell workflow that Gema started describing last week in our office hours. And furthermore, we have, from within that protocol, a quick reference card, or what we call the QRC, that I have depicted over here on the right, which is kind of like a recipe card that takes you through the salient steps to be able to complete the workflow. Here's an overview of the single-cell DNA sequencing protocol, showing both the steps that happened on last week's call and day 1, and the specific steps that I will be addressing in this presentation: library cleanup, library PCR, library purification, and then some information on sequencing. So when you get your kits, you're going to have, you'll recognize that there are multiple different storage conditions for the kits, either room temperature, 4 degrees, or -20. The reagents that you'll need for day 2 of the workflow are going to principally come from these 2 components kits: the extraction reagent from the room temperature kit, and then a number of reagents from the -20 kit. That's going to be your library master mix, your V2 index primer, and then a cleanup buffer and a cleanup enzyme. And you can certainly run a single sample at a time on the Tapestri and then process those through each of the subsequent steps that I'm going to go over on this talk. But you can also multi-sample process, meaning you can run multiple samples on the Tapestri as described in this workflow. On a, say, Monday to Thursday, and generate up to 12 individual Tapestri libraries, and then run those 12 libraries in parallel through all the subsequent steps that I will show here. So it has the flexibility to do anywhere from a single sample up to 12 samples together on day 2. We recommend if you do that, there's some guidance here about how you would separate the workflow Monday through Thursday in terms of generating all of the libraries and then pairing those all together on Friday to do the enzymatic cleanups necessary to generate a clean, suitable library for applying adapters, and then cleaning that up, analyzing it on a bioanalyzer, cooling it, and quantifying it before being ready to sequence. So the first thing that'll happen on day 2, typically you'll come in, you'll go to the thermocycler, open it up and check to make sure that your emulsions are intact. Here I have a picture of, on the left, emulsions that are not intact, and on the right, emulsions that are intact. So what you would like to see is that each of the tubes in the thermocycler has the white layer laying on top of an oil layer. The oil layer will typically be larger than when you put it in the night before, because through the thermocycling reflux process, some oil that is trapped within the emulsion layer comes out. And then when it condenses, you get a larger oil layer. So typically you'll see that. If you see the emulsions that are not intact as depicted on the left-hand side, you can contact us at Mission Bio for additional support. I'll have a slide coming up where I can show you why this might have occurred and how you could avoid it. But in most cases, you're going to get, so you'll have 8 tubes at this point after you finish the barcoding reaction that goes into your targeted PCR thermocycling reaction, typically overnight. So when you come in the next morning, you check these and you'll look for variable amounts of emulsion sitting on top of variable amounts of oil. So it's not going to be a uniform distribution across every tube. That's a typical result from a successful targeted PCR reaction. And so what you're going to do is you're going to take the green tube from the room temperature kit that is an extraction reagent and add 10 microliters of that to each of these tubes. And then in a process of adding water to them and then removing the aqueous layer and cooling those into 2 tubes, you'll go from these 8 tubes to 2 tubes, being very careful not to transfer any oil from any of these tubes to the 2 tubes that will subsequently go on for the cleanup step, because oil can impact that negatively. So here's kind of a stepwise look at what's happening during the breaking of emulsions and cooling step. So the first thing, this is just looking at one of the 8 tubes. You've got an emulsion sitting on top of the oil layer. Everything's intact, so it looks good. Then you add the extraction reagent, and you'll see that in just a matter of seconds, it'll start to break the lipid layer, and it'll start to clear. But it will not completely clear unless you vortex the emulsion. And then once you do vortex it, it'll still look cloudy. So what we recommend is centrifuging for 20 seconds and then waiting 3 minutes. And at the end of that time, if you've gotten a really well mixed sample, the emulsion will break completely and you'll have just a clear aqueous layer sitting on top of the oil layer as depicted here. But the volume of which is quite small, and it would be difficult to transfer this to a different pipette without also bringing some oil over. So we add 45 microliters of water to the top of this and then extract 42 microliters off of it so that we are able to more readily transfer the DNA in the aqueous phase without transferring any oil to our next Eppendorf tube. And I do have a bullet here to show that this is a stopping point. If you wanted to, in all likelihood, you just started this on the morning of day 2, so you wouldn't stop here. But you could also have completed this step on day 1 if you were able to finish the targeted PCR step earlier in the day. You could stop here and put your libraries at 4C for up to 24 hours or minus 20C if you want to store them for longer.
Shawn, you mentioned that was a great stopping point. Do you put that entire tube at 4C, so your library on top of the oil layer?
No, you would take the aqueous phase off and store it at that point. You don't store it with the oil. that's a good point of clarification. Yeah, you have to at least take the aqueous phase off. What we'll end up doing is combining those in 2 tubes at this point, and those 2 tubes are just the aqueous phase with soluble DNA that can be stored at -20.
Great, thank you.
Sure. And okay, so then the next step after you've combined the tubes is this 2-step cleanup process. The first of which is an enzymatic reaction. The second of which is a bead-based cleanup. That's fairly typical for library prep cleanup steps using Ampere XP beads. So the enzymatic cleanup step is you'll take the first 4 of those 8 samples were pooled into one single tube that gives you a total volume of 168 microliters. You will add 20 microliters of the DNA cleanup buffer, 12 microliters of the enzyme for a total reaction volume of 200 microliters. And you'll have 2 of these because one constitutes tubes 1 through 4 from the barcode tubes. And the second one constitutes tubes 5 through 8. And then you do a 1-hour thermocycling protocol that digests away undesired constituents and leaves us with a library that we will then bring over to clean up with the Ampere beads. These beads are standard beads that you order. You don't have to use Ampere. You can also use any other spry beads with these same criteria, and you can contact us for recommendations for suitable beads that you can use. But in any case, you want to make sure that the Ampere beads are vortexed very good right before use, because they're heavy, they can fall out of solution, and you want to make sure that you're adding the right ratio to your DNA mixes.
Shawn, I know with other protocols for using Ampere beads, they really recommend keeping them at room temp for 30 minutes prior. Have you found that to be very important?
Absolutely. Yeah, that's a great question. I have a slide coming up where I should have mentioned it here. If the beads are cold, they don't adhere DNA as well as if they are at room temperature. So we absolutely want to have them taken out at least 30 minutes before use to equilibrate to room temperature so that they have optimal DNA binding capabilities. And so here are a few best practices when working with Ampere XP beads. You're going to need ethanol for this process, and it needs to be prepared to 80% and prepared fresh with nuclease-free water, and in a manner we describe in the protocol where you'll measure out the ethanol and the water separately, combine them, and it's imperative that we do it fresh because if they've sat around for a while, that you can't be guaranteed that you still have the same percentage of ethanol or may have hygroscopically added acquired water and made the not be the right ratio. So this is imperative that we make this up right at the time that you're going to use it. Kelly's question about using the reagents and also the sample at room temperature is imperative for a good cleanup. And then also you're going to need a 1.5 mil Eppendorf magnet. Here I show one from Thermo. It's not the only one that you have to have. It's the one we recommend in our protocol, but you can contact AFAS to help you if you have another magnet in your lab that you think you'd like to use. And then once you've done the cleanup, we want to quantify and see how much DNA yield we acquired. So we would typically recommend using the qubit. We expect yields in the range of 0.2 to 4 nanograms per microliter. And you'll do this by taking 1 microliter of your purified product from your sample and using the high sensitivity reagent kit, run that on the qubit, and then record your reading for your sample. This is also a stopping point. If for some reason you need to proceed with other experiments in your lab, you can store the sample at -20 at this point. So the next step is to take Illumina adapters and apply them with a simple 10-cycle PCR step. Just as Gema explained last week, we still have the same recommendation that if you're using a thermocycler with a small number of tubes, always use the skirt or apply some tubes in the corners. Serves 2 purposes. It stops the lid from crushing the tube, which can happen and then lose the integrity of the seal and you could get some evaporative effects. And the plate skirt keeps a nice uniform distribution of heat across the entire block, which is generally designed for large numbers of samples if it's a 96-well block. So that'll keep your emulsions in the best possible state, and also your thermocycling from any evaporative effects affecting it. And so on the right, I show the thermocycling parameters for the library PCR steps. Again, it's a simple, quick 10 cycle, less than half an hour entire thermocycling protocol. And this can be done on any thermocycler that you have in your lab, ABI or any other thermocycler. Here I'm showing that the Mission Bio kit comes with enough indexes for 8 samples. They are paired I5 and I7. And in the appendix, you can find this figure that shows you the base identity for each of the I5 and I7 primers for the 8 that are supplied. And if you want to do more multiplexing than this, you can contact us and we can guide you in finding 16 additional indices so that you'll have combinations for up to 20 additional samples. And once you run that PCR step, then there's a second cleanup, which is very analogous to the one that I described earlier. You can actually use this, if you make enough ethanol in the first step, you can actually use that same ethanol for this step. It'll only be, an hour and a half later, so it should be fine. Again, having your reagents at room temperature. And now you'll need a different magnet this time. There are many different ones on the market. We have a number of them that I've used in training, and they all work suitably well. You can use these plate-based magnets, or you can use tube-based magnets. In any event, they need to be able to accommodate 0.2 mil PCR tubes. And then when this step is done, you're going to use some form of capillary electrophoresis to assess the size distribution of your library. We typically recommend using a bioanalyzer with a high sensitivity chip or a high sense chip on a tape station. But you can also use a fragment analyzer, and we can help you, guide you on the specific use of those pieces of equipment to best assess your library. Again, this is another stopping point in the protocol if you need to. Here's a typical Tapestri library size distribution for an AML library. So AML blasts run on the Tapestri, collected and then surveyed against our panel of 127 amplicons. We expect the library to be between 350 and 550 bases, the concentration to be between 2 and 20 nanograms per microliter. And if you fall within that range, you'll have more than sufficient concentration that would be required for you to sequence that library, somewhere in the range of 0.9 to 1.3 nanograms per microliter. Your plot will look something like this. We ask that you set the window from 100 to 700 and calculate the concentration of the DNA based on that field. Even though we expect the target library to reside within this range, it's possible there could be other peaks. If your library prep wasn't sufficient, you could have some dimers or other off-target products that are present that we need to account for.
Shawn.
Yep.
Oh, you're about to address my question. Go ahead.
Okay. So that was for AML. Here, I have a larger panel. This is for myeloid, and in the case where you have a more complex panel, you can get high molecular weight products as depicted here on this diagram. So I've recommended what we do in this case is and still set the window from 100 to 700 as for AML, calculate the area under the curve for that, and then also calculate the area from 700 to 1200. The most of what we are going to look for is that's going to cluster very efficiently on the flow cell is going to be in the 100 to 700 range, but we need to assess what's over that range up to about 1200 base pairs, so that because that concentration will be detected by your qubit. It doesn't cluster very efficiently on the flow cell. but we want to see how much that is. If it's a small percentage of your overall library, like less than a third, you can proceed with sequencing fine. If this is large, then I would contact a field application specialist for advice, so we can give you some protocols, basically additional cleanup steps to be able to remove the high molecular weight fraction. There'll also be the case if you have dimers in your traces. We can give you advice on how to remove those dimers with additional cleanup steps. I'll also mention that we have this quantification tool that's available. Basically, it's an Excel spreadsheet. You would put in the concentration as you just determined by the following, the description I just gave, including the average library size that you figured out. It will tell you what the concentration is in nanomoles, and then how much you take, in this case, 2 microliters of each of these 2 Tapestri libraries with this corresponding amount of water to each to produce a 5 nanomolar library that's suitable for running on a Illumina sequencer. So you don't even have to do the math. And then that'll take you to day 3 generally. Those steps will, sometime late in the day of day 2, you could put it on a sequencer on the end of day 2. That's why I'm showing this on day 3, because you probably will start it the next day. And I mentioned that our libraries are compatible currently with all Illumina sequencers. They're not all listed here. I just cherry-picked a few to show you guys the more common ones that our customers are running. Typically NextSeq, HiSeq 4000 or 6000s. So you might ask, which of these should I use? The cost is going to be a consideration, and how deeply do I need to sequence my Tapestri libraries? And so we have this guidance. Basically, it's an equation that takes into account the number of cells that you expect to get from your run, the number of amplicons on the panel that you're running, and the coverage that you desire. And our recommendation is 60 to 80x per cell per amplicon. So if we take AML as an example, this is a panel that we have built. It's one of our fixed panels. It contains 127 amplicons. And if we were to assume that we had 7,500 cells captured on our Tapestri, typical runs will result with somewhere between 5,000 and 10,000 cells in an individual run. So I'm taking 7,500 times the 127 amplicons times an average of, say, 70x coverage, and that math comes out to be 67 million reads. So then you can go to the appendix in our single-cell DNA protocol, and we have this table that is, apologize, it's very busy, so we'll just focus in on AML. So this is telling us that it's 127 amplicons. And then these are all the various different sequencers that you might decide to use, and how many libraries of AML you could pair on an individual sequencing run. And so if we say, for example, we had 67 million reads for an individual sample, and we wanted to sequence that on a NextSeq 550 in high output mode, 67 times 5 puts us in this range when we account for filtering steps that will take away some of our reads. We find that we can reliably pair 5 AML samples together on that sequencer. And if we wanted to instead run on a NovaSeq using the SP reagent kit, we could, in that case, pair 10 AML libraries together on one single run, and thereby reduce the overall cost of sequencing for the sample. And so that's all there is for the second day of the Tapestri protocol. I do want to point you guys to a new resource that we've recently launched called the Bio Knowledgebase. It's at missionbio.health. You can go there and find information on samples, sample prep, more information on the instrument itself, the experimental workflow, guidelines for sequencing, and information on our Tapestri portal, which is our software location on the web where you can launch points for the Tapestri designer, which will help you create your own custom panel. or a number of other software packages that we've created to be able to analyze single-cell DNA data, including primary data analysis, which my colleague Jose will speak about at office hours next week. This is called Tapestri DNA Pipeline. And then also a dimensionality reduction piece of software we've created called Tapestri Insights that will allow you to visualize your variants and generate subclones, and then various different visualization techniques to be able to export that data for inclusion in publications or presentations. And then also some additional tools that we recently developed using R, so that you can do more complicated analyses, including CNV and multi-omics analysis, and some of the more sophisticated figures that you might want to include, like t-SNE plots and so forth. And so lastly, I'll just point out that next week on Tuesday, Mission Bio is going to have a seminar on considerations for reopening your laboratory. This will be given by Will Oldham and Christine Scherp of Mission Bio. That's Tuesday, May 5th at 9:30 Pacific Standard Time, and again at 5:30. And then again, next Thursday, Jose will present on the topic of Tapestri Pipeline at 9:30 and again at 5:30. You can register for either of these events at the link at the bottom, and I'm now happy to answer any questions anybody may have.
Great, thank you so much, Shawn. So we had a few questions come in during your talk. What ratio of Ampere beads should I use to purify my library?
Good point. So we have slightly different ratios. The purification step is 0.72. The second one is 0.69.
Those are very precise.
Yeah.
Any tips and tricks about how to work with spry beads?
Yeah, so for sure, there's a lot of gestalt with this, right? Everybody does their techniques slightly differently. Some people keep them in the large vial that they're sold in. Some people make aliquots and then just thaw an aliquot right before they need to use it. In any instance, you want to make sure it's vortexed really well before you use it. You want to make sure that when you put the beads on the magnet, that you look at it very carefully to make sure that the beads are well adhered to the magnet, that you don't have any amber tint still left in solution, because that would indicate the beads aren't completely attached to the magnet. Generally, these beads will attach to the magnet quite readily, but depending on the dipole, depending on how strong your magnet is, it can take a little bit longer for them to apply. So we have guidelines step by step in the protocol exactly how to best handle the beads.
Great. Another question, why should I use Qubit but not the tape station to quantify my library?
So Qubit is a more quantitative measure than the tape station. Tape station is using capillary electrophoresis, which is basically using time, like retardation of migration through, think back to like a gel, although it's in a capillary. And that is less specific for concentration than the Qubit technology, which is more analogous to like looking at DNA in solution, similar to like what a Bradford assay is doing for protein. And so you get better quantitation using Qubit. That's why we recommend using that to quantify. However, the Qubit doesn't tell you, it just tells you overall concentration in solution. It doesn't really tell you if it's your desired target library or not. And so then we really need to look at the capillary electrophoresis method to see the size distribution of your library, to know that you have the desired library, that you've actually cleaned it up and it's of suitable quantity to be able to sequence it. So we really need both of those together.
Great, makes sense. Why do you have different percentage of phi-X across different sequencers?
Oh yeah, that's a good point. I did not make a point of calling that out on this table, but you can see that the phi-X ranges from generally 5 to 15 or even 20%. Those are all based on guidelines from Illumina. It's not really our specific advice. It's really coming directly from the manufacturer of the sequencer. So That's to provide sufficient base diversity to be able to sequence the library. So we go in, in accordance with what the manufacturer recommends for each of those platforms.
Great. And then I like to end with, if you could give one general piece of advice for this day 2 part of the workflow, what do you think is the most critical thing to keep track of during this part?
Yeah, I would probably say it's very early, right? It's back here all the way at the emulsion step. You want to make sure that your emulsions are handled carefully. Up through this point, if you handle the tubes in a way, drop them, expose them to shock, or they weren't thermocycled properly, and something compromises your emulsions, the quality of all the data that's generated from them downstream at that point will be compromised. So you want to be still very careful with handling the sample up through the point where you've broken the emulsions. Once you have the DNA soluble in solution, they're barcoded now, so it's essentially a bulk PCR reaction, and you can be, less cautious with the samples at that point. But this is the real point where you want to be very careful with them up through this point.
All right. Well, Shawn, that was a fantastic talk, and I know our users really appreciate all of your insights. I hope everyone out there is staying safe and busy. And Shawn, thanks again for telling us about day 2 of the Tapestri workflow.
Awesome. Thanks, Callie.